And just as a review of your DNA structure, so if we start off with a segment of DNA or your gene, you can make a chromosome by taking your DNA strand and wrapping it around histone proteins, making your chromatin, which then condense up and coil up and become your chromosome. Okay. Awesome. So let's talk about genes in a chromosome and how they code for a specific trait. Okay, so let's look here. First, we have our homologous chromosome. Remember that homologous means pairs because your chromosomes come in pairs, one from mom, one from dad. So if we zoom in into one segment or one part of your chromosome, we get what's called a gene. Okay, so here, if we zoom in, we get this one side of the DNA segment of genes. So remember, if we have one side of the DNA, we can always find the complementary side or the other side by thinking about that Chargaff's base pairing rules. Okay, so again, if we have C, we're going to get G. And then if we get G, we get C, A to T, C to G, G to C, etc. Okay, so this is what the other side would look like in our DNA strand. Okay. So from there, each gene in our DNA is going to code for a specific function in our body. And these genes will create what are called proteins, and lots of proteins will do different things in our body. And we'll talk more about protein synthesis in topic 4, but here's a little introduction. So on one single strand of chromosomes, because it's so long and has so much information, it actually codes for multiple different genes or has multiple different gene codes to make multiple different proteins. So some proteins we might see are lactase. So maybe one gene in your DNA codes for lactase, which is an enzyme that breaks down the sugar in milk. So people that are lactose intolerant actually don't produce this protein and have a hard time breaking down milk in their system or lactose, and then they get sick. Another gene can code for what's called myosin. And myosin is a protein that allows your muscles to contract. Collagen is also another type of protein that can be produced by your genes. And collagen is what gives us our structure in our skin and our cartilage. So a lot of times, when you start to get wrinkly or as you get older, your skin gets a little bit more loose and less tight. It just means you're losing your collagen in your body. And a lot of times people will start to supplement or take supplements for collagen as they get older. So fun fact, I think you start to lose your collagen at about 23 to 25. So make sure you're keeping up with that. Gene number 4, this can also be a hemoglobin. So hemoglobin is a protein found in your red blood cells, and that protein is used to transport oxygen and carry oxygen throughout your body. So these are just examples of some genes that create some examples of proteins in our body, which we'll go into more specific in terms of protein synthesis in topic 4. All right, so your DNA bases, your nitrogen bases. Remember that there are 4 of them. You don't have to memorize these. These are all found on your data sheet. But a good way to remember how they pair up, besides those little rhymes, is that guanine and adenine are going to be your big, in terms of physical structure, they're going to be your big nitrogen bases. They're categorized as what's called purines. So purines are going to be your guanines and your adenines, and they always partner up with a smaller nitrogen base in terms of size, and that's going to be your cytosine and thymine. And these smaller ones are called pyrimidines. So you have your purines, your adenine and guanine, and you have your pyrimidines, your smaller ones, which are your cytosine and thymine. Okay. So again, remember, based on Chargaff's rule, A will pair with T, G will pair with C. And then in total, when we look at that segment of nitrogen bases in a chromosome or in your DNA strand, when you add all of these together in terms of percentage-wise, they should all add up to 100 when all together added up in terms of base pairs. So here's an example. Okay, if you have a DNA molecule that has 30% adenine, how much guanine would be present? Okay, so remembering the rule, A and G are not the same, right? Remember, they're two different purines. So if we have 30% adenine, that means we're going to have 30% thymine because they pair together, they have to have the same amount. If we add that together, half your DNA molecule, or more than half, is going to be 60%. So that means the other side, or the other nitrogen bases, are going to make up 40%. So if adenine and thymine together make up 60%, that means the rest of them, guanine and cytosine, is going to make 40% of the molecule. So since guanine is the same, or not the same, but equal to the amount of cytosine, then each of the 40% will be divided in two to give you the value for both of them. So in this case, if guanine and cytosine together make up 40%, each of them are going to make 20%. So that means guanine is going to be 20% of this molecule. Okay. So there's a shortcut to solving these problems. So remember, adenine pairs with thymine. So if we have 30% adenine, we have the same amount of thymine. So that makes 60%. On the other side, we have 40% left. So that means we split that between the two remaining nitrogen bases. So guanine's going to have 20% and cytosine's also going to have 20%. So together, that's going to be 40%. And then if you add all 4 bases together, you should have 100%. But now if we look at this, remember, adenine and guanine are both purines. So that means they should make up 50% of your DNA molecule. The other 50% is going to be made up of your thymine and your cytosine, so your pyrimidines. So in this case, a shortcut is, you can always say A plus G is equal to 50. So if we rearrange this and subtracted A to the other side to have G as an unknown, you have G is equal to 50 minus A. We know A is 30%, so 50 minus 30 will give us 20% guanine. So this is a shortcut way to find individual nitrogen bases without having to find all 4 of them. Okay, so there is a DNA assignment you can do on page 33 of your workbook. And this is it right here. So yeah, take a pause in this video, or if you want to continue on, do so, but make sure you do this assignment. Okay, so moving on to DNA and its uniqueness. Okay, so now we're going to talk about how to get to DNA replication. So we know that DNA is our codes that make up everything in our body and make you, you. It's pretty much like a cookbook of instructions. So it's all the coding that decides what's going to be produced in your body. So the sequence of DNA bases determine the type of proteins that are going to be produced, right? Like we talked about lactase, we talked about myosin, we talked about collagen. So those DNA genes will tell us what kind of proteins are going to be made. Proteins are unique for each life form. So our proteins might not be the same as a plant's protein or an elephant's or a cat or a dog's. But every different species have different kind of proteins. And offsprings will look similar to parents, but not exactly the same. Remember we talked about this in topic 2 in meiosis, right? So the reason why you don't look identical to only one of your parents is that when our sex cells go through meiosis, we have new genes that are going to be combined. Or when we have fertilization, new genes are combined because a sperm and an egg with 2 different sets of genes and chromosomes come together. We have what's called mutations in DNA. So sometimes when cells are dividing, the certain number of chromosomes are not moving apart or separating properly, and then we get a mutation in either the chromosome, or maybe when the DNA is replicating, the right codes are not passed on. So then there's a change or a mutation in DNA that way. We'll talk more about that in topic 6. And then we have crossing over. So crossing over, we talked about that in topic 2, where the homologous chromosomes in meiosis will synapse and form tetrads, and then giving each other the DNA or switching DNAs within the chromosomes, and then we get new chromosomes for our gametes. So if all DNA in our body were coiled, they would stretch out and it would be able to reach the sun and back approximately 3,000 times. So that's how much DNA or how many DNA strands you have in your body. Super cool. Okay, so DNA replication. So we're going to go into the steps of how DNA replicates itself. Okay, so remember that DNA replication is necessary for one cell to become 2 identical cells during cell division, right? If we didn't replicate our DNA, the amount of chromosomes, the amount of DNA information would be lost if it wasn't replicated. So we replicate so that we have the same amount of DNA when it goes through cell division. So DNA replication occurs before cell division. So remember that happens in interphase. And if you remember specifically, it happens in S phase, right? That's where you see DNA replication. Okay. So this is what DNA replication looks like. First, we start with one DNA strand, okay? And then this DNA strand will open. It does something called unzipping. And then this DNA strand will, each side that has been unzipped is now exposed and has room for a complementary strand on each side. So now they're going to try and find the complementary side. And then in the end, we end up with 2 identical daughter DNA strands, so that are going to be identical to the parent. So both of them are going to be identical. Okay. Awesome. So identical. Cool. Okay. So semi-conservative replication. So semi-conservative, we'll talk about what that means in a second in terms of our daughter strands. But this is just another term or another way of saying DNA replication. Okay. So here, this is just a little overview. Okay. We're going to go into the steps in the next few slides. So this is just an overview of what it looks like. For a DNA replication to occur, these are the requirements. We need nucleotides, right? So remember, a nucleotide is a phosphate, sugar, and nitrogen base that acts as one unit. We need something called helicase. Helicase is gonna open up the DNA. And we need something called polymerase, which acts like a glue. So when we make up a new DNA strand, or when we go through DNA replication, we start with the parent strand, and then we end up with 2 semi-conservative DNA strands, which means that it's made up of one side that's old, which is our parent strand, and the second side is new, which is the new daughter strand. Okay. So here is step 1. Okay, well here it's just an overview again. So remember, helicases are scissors, polymerase is like our glue. Okay. So step 1, DNA, it starts to untwist. So the strand that wants to replicate starts to strain out and is initiated by something called helicase. So helicase is going to straighten out the DNA from its double helix form, its twisted form, and then it's going to cut it up. Okay, so helicase is going to unzip the hydrogen bond. So it breaks the hydrogen bonds. It goes right down the middle and cuts up the hydrogen bonds between the 2 bases. And then it opens up the 2 sides. Okay, it opens up the 2 sides of the DNA. So helicase acts like a pair of scissors to break the weak hydrogen bonds between the nitrogen bases. Okay. If you watch Big Bang Theory, here's a little meme. I wish I was a helicase. Then I can unzip your genes, but not your pants genes, your DNA genes. All right, so step 3, When the helicase opens it up, our two strands are now exposed. So the exposed nucleotides attract their complementary or their partner nucleotides. So there's what's called free-floating nucleotides. So these are free-floating in our body. Okay, so pardon my computer, it's just freaking out. So free-floating nucleotides. So that just means these are nucleotides that are kind of chilling, waiting to partner up in our bodies. And we find these from the food that we eat. So everything that we eat is made up of DNA, right? And that's going to put in more nucleotides in our body. Once these free-floating DNA or nucleotides have found their complementary partners, polymerase is going to act like a glue and fuse them together so that now we're going to start to make a new strand. Okay, so polymerase puts them together, a new hydrogen bond is formed, and then once the two strands have been formed, then we have the sugar and the phosphates of the adjacent nucleotides will then start to come together. So what happens now is our strand starts to form. We start to get a two ladder-like structure again, which the two new daughter DNA strands are going to be identical to the parents. Okay. Let's see. So in the end, when we finish our DNA replication, we get two new daughter strands. These are called semi-conservative. So semi meaning half, and then conservative meaning old. Okay, think of like a conservative idea as like an older idea. So half old, which means it is half from the parent strand. So the original strand, the OG, and then half is going to be the new strand. So this is the new daughter side. So that's why it's semi-conservative, because it's half old and the other half is new. So we get two semi-conservative DNA strands in the end. Okay, so that's how you get DNA replication. Now, there's always going to be mistakes in replication, which will cause different proteins to be produced or mutated proteins. So mistakes in replication can be caused by environmental factors such as hazardous chemicals and radiation. So when you're exposed to things that your body shouldn't be in terms of harmful products, this can actually change your DNA. So think of it like cancer, right? Or any sort of mutation disease. So one change in DNA sequence can have a significant effect. So if you see here, strand A, that's the original strand, if it replicates and for some reason in strand B, instead of C bonding with another G, which is supposed to, there might be a mutation where A sneaks in there and then cause a mutation here. Okay. Now you might not see it on the surface, so here is an example of a plant. Kind of looks like a mint plant, but here, even though they don't look different, what can happen is that the difference is one plant is susceptible or resistant to herbicides. So it's a resistant plant. So if you put a herbicide on this mint plant, because mint plant can be very invasive. Mint plant can take over your garden. They're weed essentially. And if you're trying to get rid of it, you might spray some herbicide onto it and usually it would die. But if there's a mutation, it might cause that plant to be resistant to the herbicide and it wouldn't actually die. So in this case, this might be caused by a DNA mutation. Okay. So in the end, in mistakes, there are approximately 3 billion bases in our DNA. So mistakes, which means faulty base pairing, so the nitrogen bases are not paired with the right nitrogen base, are infrequent and can permanently damage. Mistakes, faulty pairing, are infrequent and permanent damage prevented by proofreaders, so enzymes. So even though our DNA can cause or can have mistakes in them, our body actually has what's called have the enzymes that can actually find these mistakes and fix it if the mistake is minor and if it doesn't occur very often. So these enzymes run along DNA and they look for mismatched pairs. So they look for nitrogen-based pairs that aren't supposed to be together. And some of them are able to snip out the errors and replace them with the correct sequence. In that case, our body just naturally fixes its own mutation. But if you have a mutation that is reoccurring or is very large, sometimes these enzymes don't actually work, and then you create a faulty or mutated protein. Okay. To end here, we're going to talk about the Human Genome Project. And all the Human Genome Project is this project back in the 1990s that identified all the genes in human DNA. So scientists were able to locate and find every single base pairing, every single gene in human DNA and map it out. Okay, so it was completed back in 2003. So it started in 1990, and then by 2003, they had a final mapping of all of it. Okay, so here we go. So in 1990 is when they first started. Here's just a little timeline. You can see this in your notes. And then it continues on. And by 2003, there's a finished version of the sequence. So now we know every single gene that's available in humans. Okay, so what's good about this is that you can actually use it to compare people's genes to like a blueprint pretty much. And you can see like if the blueprint matches the person's genes you're looking at, then they're probably healthy. If there's something that's different, maybe you can diagnose any sort of genetic disorder. From that, you can probably develop new medications. You can prevent any disorders from occurring. And pretty much in the end, you can identify all DNA. Some issues with this is, well, now we have a blueprint of everyone's DNA. Who should be able to have it, right? Like, should your companies have it? Should your employers have it? Like, who can have access to this? In this case, how would we use the data for reproductive reasonings, right? Like, are we going to use this now to like fix different genes if we know that a certain gene does something to someone to cause some sort of disease? This leads into then designer babies and all of that, right? And then in the end, how much is this going to cost society? So these are all different perspectives, right? So cultural perspectives, societal perspectives, economic perspectives, etc. Okay. Here's just some extra information for you guys, some examples. Angelina Jolie had a mastectomy. So she got her breast taken out so that she can prevent any sort of breast cancer from developing, because her family had a line of history of breast cancer. Her mother had it and her grandmother had it. So in this case, to prevent that, she knew that she had the faulty gene of BRCA1, which then would cause breast cancer or start breast cancer in her genes. And then in this case, she prevented that from happening by having a mastectomy. Okay. And that's it. So that's topic 3. Thanks for watching.